2015, Pavlo was recruited in our department, and he gave a talk about data dev, some version of it, and I thought, okay, this is a very interesting topic, and I tried to relate this topic with a question I had 2 years before, when I was spending my sabbatical leave in the School of Psychiatry at UNSW Sydney. So I spent this 1 year sabbatical leave there, and I had some interesting questions, and I'm going to introduce these questions now. And Miriam is still working in NSAI, and Pavlo now moved to Telecom Paris. So let me start with this origin of this work. So as I mentioned, I spent 1 year working with these people at the Center for Healthy Brain Aging at UNSW Sydney, and they had this interesting old Australian twin study where they measured a lot of variables on old twins, like age, sex, kind of information, and stuff like that. And in this work, I was interested in relating genetic information to neuro images, right? And for this specific work, the kind of genetic information that was interesting to us was called the zygosity, which is the fact to know if the twins are identical twins or non-identical twins. All right, so in the brain you can measure different things. There is the gray matter and there is the white matter. So the gray matter is, these are the small cells which process the information, if you want. And the white matter, these are the cells that connect, pieces of cells that connect one neuron to another group of neurons. And these are called axonal fibers, and they are white because there is some molecules around that are called myelin, which is white. Okay, and this is an interesting question to understand if there is some genetic background in this white matter integrity, because if genes are involved, then you can hope to try to cure or improve disease such as Alzheimer or this other disease I mentioned here, by looking at what are the genes involved. And if this is not related to genetics, then maybe you want to find what are the environmental factors that are involved and try to do some prevention. So how do you measure these kinds of white matter in the brain? Well, these fibers in the brain are surrounded by water molecules, and the water molecules, they diffuse more easily along the fibers that perpendicular to these fibers. And so if you use a scanner, then you can see this water molecules displacement, right? And then there are some mathematical models to try to model these water molecules displacement. This is not the topic of today. And then we use something called tractography to obtain measurements of these fibers. So you can see here that there is a main fiber tract in the brain, which is a fiber tract that goes from the brainstem to the motor cortex, and which is present in all mammals, because this is the fibers, the nerves that allow us, for example, to move our hands or legs and so on, right? So we are interested in this main fiber tract, and these are examples of data that were collected on different subjects. So you can see on the left and on the right hemispheres, we measured 1,000 fibers, right? And we need some new statistical modeling strategies to be able to model to model these interesting type of data. All right, so I mentioned that Pavlo was working on data depth. So what he presented was something related to the original concept of data depth. Right? So this concept was introduced by Tukey in 1975, and he introduced this notion of depth of a point with respect to a cloud of points. And this notion can be extended to the depth of a point with respect to a probability distribution. So you can see here on the first the first formula that if we are interested in computing the depth of this point x in red with respect to a probability distribution Q, then we just have to compute the infimum of Q of H. So we compute the probability of H, where H is a closed half space that contains our point X, right? So this is the infimum of all the Q of H. And in fact, we can rewrite this formula by considering all spaces HUX. And this HUX, these are the closed half spaces that whose frontier contains X and whose frontier is orthogonal to a vector U, which is on the unit sphere, right? And the formula at the bottom is the empirical counterpart of this first formula, meaning that if we have a set of observations, x1 to xn, that comes from this distribution Q, then we can compute the half space depth of this point x with respect to this multivariate cloud of points xn using this last formula in blue on the bottom of the slide. All right. So let me maybe introduce a simple example where we try to understand a bit better this formula. So here we have two variables, age as a function of versus weight, right? And I put back this formula at the top of the slide. So we have 161 points, and we want to compute the depth of this point here in red. So what is the depth of this point with respect to all the points here in this two-dimensional plot, right? So what we do, we consider this half space here, and we consider the proportion of points that are contained in this half space. So it's 120 divided by 161. And we want to compute the infimum of these proportions for all half spaces that we can consider. So here I consider another half space, and the proportion of points is 112 divided by 161, and so on. Okay, so we consider all half spaces, and the infimum is obtained here. So 13 divided by 161. So this infimum and this proportion is the depth of our red point with respect to the cloud of points, right? So this is the depth of the point. So now if we consider another point, which is maybe more on the outside of the cloud of points, so these new point in red here, we can do the same thing and compute the half space depth for this point, which is now 3 divided by 161. So you can see that this depth is smaller, which indicates that the point is more on the outside of the cloud of points. So this is this original notion of depth introduced by Tukey. You can also compute Tukey trimmed regions. So to do that, you consider, for example, when alpha here is fixed to 2 divided by 161, then you compute all half spaces that contain only 2 observations, right? And you do that, and then you obtain your trim region for this value of alpha. And of course, you can do that for any kind of value of alpha, and you obtain something like that. Okay, so now our brain fibers, they do not live in a 2-dimensional space, they live in a 3-dimensional space. Okay, so of course, what I just presented can be generalized to a three-dimensional space. So it's exactly the same formula as before, but now, of course, this hyperspace, this closed half space, is defined with observation in R to the power of 3. And S is the unit sphere in R3, right? And we can visualize that on this figure here. So you see here in the middle, this is the unit sphere. This U here is one element on the unit sphere. And then we have our point X here that we are interested in. And we can consider this hyperplane that goes through X and which is orthogonal to U. And then H of Ux is the closed half space on the right of this red hyperplane. And of course, we let U move on the hypersphere. So we will consider all the possible U's on this hypersphere and then all associated HUX, corresponding HUX. All right, and we can construct also these depth regions in 3 dimensions. That's exactly the same idea. All right. So, so can we use this concept to try to do something with these brain fibers? That was the original idea I had when I saw Pavlo's talk in 2015. But then we realized that the brain fibers, even if they are only observed at a finite number of points, these points are connected. So if we apply directly this original notion of depth to these fibers, which consist of points connected points, then you lose the links between these points. Okay, so can we use a more advanced concept of depth? So in fact, there is a depth for functional data. So here you can see functions, right? And the formula in the middle is the depth of a function y, which is one realization among these functions. And how it is constructed? Well, you just take a vertical, all right? So for t that goes from 0 to 1, if you fix the value of t, then you fix a vertical at this value of t, and then you consider all the points on this vertical. And then you compute the one-dimensional Tukey version of the points on this vertical. And then you do the same thing for every vertical, and you integrate for all the verticals. And then you obtain this depth version for functional data, right? So if we do that for these curves here, these functions, then this is what we obtain, right? So we can compute the depth of each curve with respect to all the other curves, and then we can give a color value to these results. And for example, the blue one is the curve that obtained the smallest possible value of depth with respect to the other curves, right? So this is the most central curve, if you want, and the yellow curves are the most outside curves with respect to these values of depth that we just computed using that formula. But now our brain fibers, they live in a three-dimensional space, okay? So they are three-dimensional functions, so multivariate processes, okay? But in 2014, Klaskens and some other people, they developed an extension of this notion of depth for functional data. So it's called multivariate functional half space depth, and it's exactly the same idea, right? So you fix a value of t, and then you cut a slice in the space, and then you compute the classical original version of Tukey, and then you integrate for all values of t. And this is what you get for multivariate functional half depths, right? But is it a good idea in our context to use these versions of functional half depth? I'm going to show you an example where we consider two different parameterizations of an object which is like that. So it's a curve which is a S-shaped curve, and we consider these two different parameterizations of the same object. And here you can see this object here, and if we look at this first parameterization, you can see that the kind of particle goes very slowly at the beginning of the curve and then very fast at the end, all right? But now if we consider the second parameterization, then it goes very fast at the beginning and then very slowly at the end, all right? So it's two different parameterizations of exactly the same object, all right? So two different parameterization of the same object. So now, if we apply these functional or multivariate functional depth to these set of curves, so here I consider 100 S-shaped curves, and I parameterize these curves using parameterization A on left or parameterization B on right, and then I apply this notion of multivariate functional depth. And the results are on the two graphs at the bottom. And you can see that, for example, the blue curve, which is the more central curve, is different depending on the parameterization. Okay, so it means that different parameterizations lead to different results when we use this concept of multivariate functional depth. This is another example, a classical example in functional data analysis. So we have these two sets of curves parameterized either by time or by length, by arc length. And in this table, we give the rankings that we obtained. And you can see here, for example, we get a ranking of 1 when we parameterize by time, and a ranking of 13 when we parameterize by the arc length. So very different results depending on the parameterization. Okay, and this is not what we want for our specific application, because this curve that we observe in the brain, they don't have some specific kind of parameterization, intrinsic kind of parameterization. All right, so now I need to introduce a few mathematical concepts and notation to be able to introduce our generalization of this notion of depth for curves that get rid of this parameterization problem that I just mentioned. So a path or a parameterized curve is just a continuous map from 0, 1 to r to the power of d. S gamma is the locus of gamma. So it's just the image of gamma. Okay, so we see this segment 0, 1, and for each element in 0, 1, there is a corresponding point on this curve here. And because we want to get rid of the parameterization, we are going to use an equivalence relation on curves. And we are going to say that two paths, gamma 1 and gamma 2, are equivalent. And we note that gamma 1 are gamma 2 if they share the same locus and if the points are visited in the same order. So in my previous example, this example, then these two parameterization that I showed you, the red or the blue, these are two parameterizations that give the same locus and the points are visited in the same order. So these two paths are equivalent. All right. So then we define the an unparameterized curve. This is just the equivalence class of gamma up to the equivalence relation that I just introduced. Okay, so somehow we put all the curves that are related one to each other with this equivalence relation in the same bag. Right? And then we define the space of unparameterized curves here, which is just the space of these equivalence classes. And if we endow this space with the Fréchet metric, which is defined here, then this space becomes a metric space, and thanks to these nice properties, we can define probability measures on this space. All right, so now we need to introduce the length of a curve. So how do we do that? The length of the curve, you can see here, we subdivide the curve in small segments, and then we just sum the length of these segments, right? So we partition the curve in small segments, and we sum the length of these segments, and we try to compute the supremum of this length. So of course, if the partition is becoming, the grid is becoming smaller and smaller, so a finer grid, then we increase the length, and at the limit we obtain, we can estimate or approximate the length of this curve. So this is our definition of the length of the curve, and this is independent of the parameterization. So it's just the length of the unparameterized curve. All right, so there is also this normal parameterization, and in this proposition in 2006, we can see that any curve, any unparameterized curve can be uniquely parameterized using this so-called arc length parameterization. All right, and this arc length parameterization is just that we move at constant speed along this curve. All right, so this would be a different parameterization of this S that I showed you. It would consist in taking a parameterization where the particle will move at constant speed along this S-shaped curve. All right, so the last thing we need to introduce is the line integral along a curve. So the line integral of the function f along this curve, unparameterized curve C, is defined by this formula in the middle. Right? It's just the Riemann integral of f of beta c of t, where beta c is just the normal parameterization that I just introduced, multiplied by the length of this curve. And now we can introduce for every Borelian set A the mu c of A, right? So the probability measure mu c we introduced is just the line integral of the indicator of the set A divided by the length of C. So roughly speaking, mu c of A is just can be interpreted as a portion of the length of the curve C that intersects with this set A. So now we have all the ingredients to be able to define this notion of depth. So I start with the depth of a curve C with respect to a probability measure P. Right? So this probability measure P is a probability that belongs to the set of all probability measures defined on the set of unparameterized curves, right? Which have a finite length, right? And here I denote by key a random element on this space with distribution P, right? So random element key. So you know, a random variable is just an object that produces real values. Here, this random element, it does not produce real values. It produces curves, right? Unparameterized curves. And the probability to produce these curves is defined by P. And so now we define the QP of A by this formula. So it's just the expected value of mu key of A. Right? Mu key of A, what is it? Remember, so key is a random element that produces a curve C, and mu C of A is just this formula here. So it's just how a curve intersects with A. So here is just, on average, how all these curves generated by key intersect with A for a given A. Right? And now we can define this formula at the very bottom of the slide. So remember, we computed the depth for a point with respect to a cloud of points or with respect to a probability that produces these points by the infimum for all these half spaces. So here it's a bit similar. So it's the infimum for all u that belongs to this hypersphere of this ratio. And this is for point x, which belongs to the locus of points. So for each x that belongs to the locus of points, so for example, here for each point that would belong to this shape, we would compute this formula. And then we will integrate here for all points on this locus, this formula here. And this formula here will give us the depth of the curves, unparameterized curves, curve C, with respect to the probability P. There is a sample version of this curve, and the sample version is we just replace in this formula here the measure QP by the empirical measure QN. So QN is just the empirical counterpart of the QP measure, and we plug this empirical measure in this formula, and we obtain the sample curve, the depth of the sample curve depth, sorry, of C with respect to a sample of random curves K1 to Kn, right? So maybe we can spend 1 minute on trying to understand the rationale behind this formula. So this formula that I just introduced, so this formula which is here, because beta C is the normal parameterization, it's not, and Qn is defined like that, it's not very difficult to see that we can rewrite this formula like this, right? And now we want to compute the infimum of this formula. So this is minimum when the denominator here is large and the numerator is small, right? So we want mu C to be large, and we want this part at the top to be small. Now, what is mu C of H? H is one of the hyperplanes here, right? So for each u here, we got a corresponding hyperplane, and we will consider all the hyperplanes. So let's say I fix this first hyperplane here in green. Mu C of H is just how much the curve C intersect with H. I can go back to this formula I mentioned here. Mu C of A is how much the curve intersect with A, right? So here, mu C of H is how much the curve C intersect with H. So here, the C curve is in red. So how much the C curve in red intersect with H. So this is the fraction of the length of this red curve that goes into this HUX. Okay, so this part of the curve. And now at the top here, I do the same job for all the other curves, C1, Cn, all these other blue curves. And I compute, you see, the sum divided by n, so I compute the average. So on average, how much the blue curves go into this green space. And I take this ratio. And I do that for all the possible green spaces, all the possible half spaces. So the infimum will occur when the red curve will be quite different to the blue curves. And when it's different, then we will be able to find a green space, a green plane, where the red curve enters deeply, so mu C will be large, but the blue curves do not enter deeply, so the numerator will be small. Okay, and if this is the case, then this measure will be small if we integrate for all the values. Okay, so this is the rationale, the intuition behind this formula that we introduced. So we proved a number of results for this new notion of depth for curves, for unparameterized curves. So we proved that this depth is always between 0 and 1. So it is 0 if the depth is small, so it means meaning that there is an outlier or the curve is outside the bundle of the other curves. It's 1 if it's right in the middle, okay? So like the median, for example, right in the middle. Similarity invariance. So it's invariant to a rotation of the set of curves or scaling of the set of curves, and vanishing at infinity. So if we take this red curve and we send this red curve very far away from the main set of curves, then its depth will tend to 0. It will converge to 0. And we proved also the consistency of the empirical version. So the sample version of our curve depth will converge to the population version when we increase the sample size. All right, so computing this integral can be a difficult task, right? Even if we have observed all the C1, Cn, and we know C, computing this infimum over all possible U's on a hypersphere is a difficult task, and then we have to compute the integral over this infimum. Okay, so this is not so easy, and it's only possible to obtain a closed form expression in very simple examples. So to help people to use this measure, we produce here a Monte Carlo algorithm. Okay, so this algorithm, it has several steps. So the first step here, step 2, we generate observations, and these observations are used to estimate mu c. So these are used to estimate the denominator here. Then we generate from step 3 to 6, we generate uniformly observations along each curve, and we use that to estimate the numerator here. Okay, then from step 8 to 10, we compute this infimum. All right, so we take the smallest ratio for all these points, and as points we consider these points Z1 and Zn, and we also use these points Z1 and Zn to estimate this integral here from 0 to 1. Okay, so I can show you an example here. So we want to estimate this big formula, and we are going to consider this very simple example where we have 25 concentric circles, and we are going to consider our algorithm generating only m points, just to be able to see what's happening. And we want to compute the depth of one circle with respect to the other circles. So this is what we get. Okay, so we generate yellow points here to get the integral, red points along this red curve C to get mu c, and blue points along each other curves to compute the numerator. And then we consider all these, we consider all these point U on the hypersphere, and then we compute the infimum for all these half spaces HU, right? And if we do that, then this is what we get, right? So if we compute this depth of each circle with respect to all the other circles in this set of 25 circles, and then we assign a color to the depth that we obtain, then, for example, the blue curve here is the curve that is detected as or labeled as the most central curve, and the yellow curves are the most outside curves. So it shows that our procedure, the Monte Carlo procedure, and this definition is working, behaving properly. Right? And in the paper that is associated to this talk, we have explicit expressions for this quantity for this very simple example of curves. And so we can see that we are doing a good job. Now I'm going to apply these results to 2 applications. So the first one is hurricane tracks. So in this paper that was published in 2014, these authors, they proposed a visualization of several historic European cracks in the Gulf of Mexico between 1920 and 2012. Right? So you can see on this graph at the bottom that the results they obtained using some sort of modification of functional depth is not very satisfactory, in fact, because the red curves here, these are labeled as outliers. But you can see that, for example, this curve here is right in the middle, quite close to the yellow curve. So the yellow curve is the more central one, and this red curve is labeled as an outlier. And it's strange to think of this curve as an outlier because it seems to be quite in the middle. Okay, these curves could be an outlier, but this one it's a bit strange. All right, and this one it's a bit strange also. So if you are interested in this type of data, you can go on this website here, and you can download and click on this map and download a lot of curves like that. So we did that, in fact, and then we applied various procedures on this curve. So here, this is the multivariate functional half-depths that are presented at the beginning of the talk. And you can see here, using this time parameterization, then there is the same type of problem. Okay, these red curves, these are identified as outliers, but they are right in the middle of the bundles, so they should not be labeled as outliers. If we use an arc length parameterization, we can see this is the same type of problem that occurs. If we use another well-known depth method, which is called the multivariate simplicial depth, then we get the same type of problems with time parameterization or with an arc length parameterization. And then we applied our new developments, and this is what we get. Okay, so now the most central curve is here, right in the middle of the bundle, and the red curves, which are labeled as red or colored as red, thanks to the findings of our measure. These are outliers, and these results are meaningful. All right, so this approach could provide maybe better confidence of the US regions that are more at risk of suffering of a hurricane. Okay, this could have some potential interesting applications for insurance companies. Now, let's get back to this application to the brain fibers. So the original motivation for this work. So remember, we have this older Australian twins study that includes this DTI data, so diffusion tensor magnetic resonance images. So we consider here 34 twins, pairs of twins, so 68 twins. And among these twins, we have the DZ twins. So these are the non-identical twins, and 23 MZ, so the identical twins. And for each one of these twins, we extracted 1,000 fibers connecting the motor cortex to the brainstem for the two hemispheres. And the various questions that we would like to address in this work are in terms of information compression, so for better understanding of the brain organization in terms of this white matter. The outlier detection, so can we detect some fibers that are wrongly tracked? Curve registration, so this is important to align two brains together, because when you are in the scanner, you can have your head a bit on the left, a bit of the right, so you want to realign this set of fibers together before doing some further analysis. And then to study the genetic dependency to identify some potential cause of disease. So these are the data that we have, and we apply this notion of this that we developed, and this is what we get, right? So you can see here that we give a color depending on the value of depth that is found. Okay, so each curve in the left bundle of fibers, for example, we compute its depth with respect to all the other curves in this bundle. And we do the same thing on the right hemisphere. And then we can decide to, for example, display only the curves that are right in the middle, that have a value of depth that is above 0.624, for example. And by doing that, or we can add some transparency also. So by doing that, it makes easier for neuroscientists to visualize these fibers and to understand what are the possible differences, identify some outliers and things like that. Okay, so if I keep only the most central fibers, then of course I'm doing some information compression. Okay, so this is the median of the bundle, if you want. In terms of outlier detection, this is what we get. Okay, so we put in red the fibers that obtain a very low value of depth. And you can see that these red curves, they are correctly identified by our method as outliers. We compared this ability of detect outliers to the other well-known depth methodologies, like the multivariate band depth, simplicial band depth, and these other methods. And the last one is our curve depth. And you can see here that, for example, for this SBD, this curve here is not detected as an outlier. So it doesn't do a good job in that respect. Here you can see that we have a lot of red curves in the middle of the bundle, right? So we detect as outliers curves that are in fact in the middle. And for our proposal here, you can see that there is a small gap here that help us to identify, okay, these are the guys that are outliers and probably these are not outliers. So it's a very steep increase here that help us to identify nicely outliers. Okay, this other application is in terms of curve registration. So what we want to do, because two persons can have different brains, different sizes of brains, right? You have some person that have a brain which is big. Some people have a small head, so a small brain. Doesn't mean anything in terms of intelligence, of course, but we want to be able to align these fibers together, right? So what we did is the following. For each subject, let's say we consider only the 1,000 fibers in the left hemisphere. So for each subject, we compute the most central curve for the bundle of fibers of this specific subject. Okay, so the fiber that has the highest value of depth. Right? And we do that for every individual, every subject. So we obtain one more central fiber for every subject. We put all these 68 more central fibers together, and then we compute the most central of all, the deepest of all. And the deepest of all, this is this fiber which is here, the dark blue fiber. So this was obtained as the most central fiber for subject 206 and 35, in fact. Okay, this is a code for the 68 people, so we cannot identify them for ethical reasons. Then when we have this deepest of all, then we try to move the deepest fiber of every subject closer to this deepest of all fiber. Okay, so we do that by using a least square approach, where we try all the possible rotations and translation to bring closer these deepest fiber. So the red curve here, for example, is the deepest fiber of subject 104, and we try to put this red curve as close as possible to the dark blue, and the result that we obtain is this one, okay, the light blue. And we do that for every subject. And you can see here the distances before and after this process. So 10 before and 3 after for subject 104 and so on. So we are able to realign all these curves together. So when we have done that job to try to make all these brains comparable in some sense, then this is what we have done. Okay, we computed these plots which are called DD plots. So here, for example, a red dot here, a red circle here, is a fiber for subject 105. So, okay, let's consider two subjects, two twins, 105 and 205. So these are two twins, DZ twins, or non-identical twins. So now I consider one fiber of the twins of the twin 105, and I compute its depth with respect to itself and with respect to the other twin. So I take this fiber, I leave it in the bundle of fibers of itself, and I compute the depth of this fiber with respect to all the other fibers of the given twin, and I obtain a 0.6. Now I take this fiber and I put this fiber in the set of fibers of the other twin, and I compute the depth, and I obtain a value here. And I do that for all the fibers from one twin with respect to himself, and from this twin with respect to the other twin, with the fibers to the other twin. And then I plot this thing. And I do the same thing for all my pairs of twins. And you can see on the first row, these are non-identical twins. For the second row, these are identical twins. And if the distribution is exactly the same, then all the points should be aligned on this black line in the middle. Right? Then we computed p-values associated to that. So this is some sort of generalization of the Wilcoxon test based on ranks. And the p-values are in the caption here. Okay, so here we have a very small p-value, very small p-value, very small p-value, meaning that we reject the hypothesis of equality of distribution of the spatial organization of the fibers. And here we have 0.733, 0.366, 0.366, so large p-values. So now we do not reject the hypothesis. So it means that we can show that the non-identical twins have a different distribution in terms of organization of the fibers, but the identical twins, they tend to have the same type of organization. Okay, so it means that the genetics drives the spatial organization of the fibers. So as a conclusion, what we obtain is, what we built is a novel notion of data depth that can operate on the space of unparameterized curves. It has some nice properties, like mentioned, that similarity invariance, translation invariance, rotation invariance. It only exploits the geometry of the data, and it's invariant with respect to any kind of parameterization of the curves. And we have other examples, applications in the paper in terms of unsupervised or supervised classification. And we have this R package curve depth on the CRAN that is available now if you want to use that. And as a final note, so these results I mentioned, we obtained that on 68 people, but now we are working on 40,000 brains, and we are using this supercomputer GADI that is available in Australia to obtain all these results. So thank you very much for your attention. I know the slides are recorded, so I show you the references if you want to go further. So now I'm happy to answer your questions if you have some, and I think Hassan could unmute the microphones maybe. Thank you.
Thank you very much, Pierre. It was a very interesting topic. I like that you use depth curve for comparison of the distribution of two kind of actually twins. That was interesting. So any question, David?
Yeah, I really enjoyed the talk. Thanks. I wasn't familiar with this idea of data depth. It seems that in the notorious, n very much smaller than p problems like you get in bioinformatics, that this notion might not be much good, because in general you'd expect the entire sample to be a simplex in the high dimensional space, in the p-dimensional space. But in this case, by integrating, you've managed to convert what's really a very high dimensional data set into a sequence of three-dimensional data sets, and thereby retrieve the utility of the data depth concept. And I'm just wondering, do you have any idea of how you can do something similar for, say, gene expression data where p is very much larger than n, but there's not the same sort of geometry?
Yeah, that's a very interesting question. I'm trying to retry this name. Yeah, there is this guy this French guy that got this field medal quite recently, and he was working on some notion, but I can't remember what that is. But anyway, this is basically a way to from what I understood, because it's a difficult topic, it's a way to have some sort of one-to-one correspondence between a curve and a point in a very high dimensional space. So yeah, that was an idea that we wanted to explore, in fact, because this notion of depth, it's not possible to use that when you have when p is very large. It's too time consuming, in fact. So that was an idea to try to take these points when p is very large, send that using this approach by this guy. Maybe the name will get back to me, and then use our notion of depth to compute the depth, basically.
Yeah, sounds intriguing.
Yeah.
Okay, thanks.
Yeah, you're welcome.
Any other question? You are allowed to unmute yourself at the moment. Any other question? Can I ask my question as well?
Yeah, sure.
Is there any room to improve your method? I mean, for future research, what's your idea? If someone like to improve the method, is there any room or possibility or disadvantage that we can try that?
Well, you see, we built this idea using this ratio of two quantities. So we had to put some conditions, because sometimes when the denominator of the ratio is small, this can create some problems. So maybe that's something that needs to be studied a little bit more. Also, we decided to look at this ratio, but maybe there are some other ideas to consider instead of ratios. Also, for this example, for example, I mentioned for the circles, then for circles, it's not a good idea to use hyperplanes, because hyperplanes, the frontier is just a, yeah, kind of a line, right, in two-dimensional space, and so it's difficult to grab the essence of the organization of the space. So using some circles instead as frontiers would be more interesting, like using balls, for example, of increasing sizes as kind of hyperspaces would be an interesting thing to do, maybe, but maybe then the formulas might be more difficult to get. Yes.
Wow, thank you. Thank you. Any other question? maybe I can ask my second question.
Sure, sure.
In slide 40, in that slide, I'm wondering how you compute p-value.
Okay, it's just if you look at the Wilcoxon rank test.
Okay, this is asymptotic normal distribution.
Yes,
for,
yes, exactly. Yes, yes, yes.
Okay.
Yeah, this is what we do. Yes.
Okay, thank you very much. Okay, I think we don't have any other question. Thank you very much for your time and everyone for joining us.
Thank you very much.
Here.
Have a nice day, everybody.
Thank you. Have a lovely weekend. Bye-bye.
Thank you. Bye-bye.